Hi, everyone. Good afternoon, and thank you for joining our first event this year. For those who are new, Research and Diversity, R&D, is dedicated to building a community for underrepresented minorities in STEM. By speaking with experienced researchers about their accomplishments, we hope to inspire STEM students beginning their research careers and highlight potential role models for them. Just as a heads up, we are recording the interview, and we'll post it on the website after, like I said. And if you'd like to ask a question, please message me directly in the chat and I'll hop in and make sure that you're able to ask. Some of them we may end up waiting towards the end because we might end up having similar questions. My name is Erica. I'm originally from North Bergen, New Jersey. I'm a junior at General Studies, studying biology. They didn't have micro. I'm the head of development and a co-founder of R&D.
My name is Shamar, and I'm from Brooklyn. I'm a senior in C studying biomedical engineering, and I'm also a co-founder and the head of technology and design.
And I'm Vivian. I'm a sophomore in Columbia College studying physics, and I'm also the head of outreach and a co-founder of R&D. And today we'll be talking to Dr. Amanda Brown, who is an associate professor in the Department of Neurology at Johns Hopkins University School of Medicine. Her current research interests, funded by the National Institute of Neurological Disorders and Stroke, are centered on identifying the mechanisms and consequences of HIV-induced pro-inflammatory signaling in the central nervous system. Her lab is currently using humanized mice and in vitro approaches to identify the critical cellular pathways and the molecular mechanisms involved in HIV macrophage biology and persistent and latent infection of those cells. Dr. Brown serves for the following NIMH or NIMDS funded programs as co-director for the developmental core of the Johns Hopkins Center for Novel Neurotherapeutics, which promotes innovative translational research projects and to promote diversity in the neuroscience workforce. She is co-director of the Translational Research in Neuro-HIV and Mental Health, director of the Johns Hopkins Internship in Brain Science Program, director of the Johns Hopkins Neuroscience Scholars Program, and director of the Johns Hopkins Neuro-HIV Comorbidity Scholars Program.
For this interview, we will be discussing the role of osteopontin, or OPN, in regulating inflammatory signaling pathways and function in macrophages, and uncovering molecular mechanisms by which OPN stimulates HIV replication in this cell type. OPN is upregulated in several neurodegenerative diseases, including HIV-associated neurocognitive disorders, also known as HAND. However, its role in these pathogenic processes is not fully understood, and studies are underway to begin to fill this gap in knowledge. Dr. Brown, would you care to tell us more about it?
Yes, thank you so much. R&D, what a great name. Thank you so much for the invitation. It's a pleasure to be here. And as you were reading off all those titles, that's just to give you a hint. Being at a soft money institution, right, that I get a little bit of percentage in terms of support for my work from each of those activities that I'm sure we'll get a chance to talk about. Okay, so should I share my screen? I didn't know if you wanted to have slides or not. We don't have to. We can certainly start without them. And so let's just go ahead and do that. So being in your position, I always would look at faculty and I'm like, how do they get to where they got to? They seem to have it all together. So it's very interesting, my training pathway. I actually didn't train in neuroscience, but during graduate school, I got interested in how pathogens evade the immune system, and specifically intracellular pathogens like tuberculosis infect macrophages. And interestingly, HIV, the human immunodeficiency virus, also infects tissue macrophages. And at the time, there wasn't a whole lot known beyond that. And so that's really where I focused my research. And my mentor actually encouraged me to study HIV macrophage biology. And in so doing, I developed a novel tool, which at that time was a novel tool, which was to insert the green fluorescent protein into the early gene position in the HIV genome. And what this allows us to do is when the virus successfully infects the macrophages, we could use a variety of fluorescent-based techniques, flow cytometry, microscopy, to actually identify and isolate the infected macrophages. And so I was asking the question, what are the whole cell genes that are upregulated as a result of HIV infection of macrophages? And this is how I discovered osteopontin. At the time, there were only 2 papers reported. There was a paper in humans, HIV-infected humans, showing that osteopontin was elevated in the lymph nodes, in a particular type of lymph nodes. And there was another study using a monkey model of HIV infection, showing that osteopontin was increased. And so based on those 2 studies, I thought, this is probably a good protein to track down, to figure out at the mechanistic level, how does it function? What is it doing? And so that then led to a whole series of papers where we've tried to do that. And so I'll just jump ahead, and because I know we have limited time and you guys have questions. For most of my career, most of the papers that you'll see from 2011, it was 2011, we reported the first discovery of osteopontin being elevated in HIV infection after another group, Trish Bordeaux's group, in 2008. But it's only in the last 4 years or so, using my first R01 grant to actually use a mouse model to ask the question, is osteopontin required for HIV-mediated neuronal injury? So I'm skipping a lot, but in the introduction, you heard that HIV, because it infects tissue macrophages, these types of macrophages are in every part of the body, actually. And in the brain, we have several types, including a specialized one called the microglia. And microglia are the immune cells of the brain, but they also perform very important functions for neurons. So HIV infecting these cells causes an activation response, which results in the release of cytokines, chemokines, and other factors. And people have shown over the years, you can get dysregulation of reactive oxygen species, of metabolism, and again, this neural exacerbation of neural inflammation. And so most recently, we've used the humanized mice model. So normally mice cannot be infected with HIV, but since the 1960s, 70s, for different purposes, really from the cancer field to help with studies of xenotransplantation, people have been trying to make better and better immunodeficient models in which we can then reconstitute the models or reconstitute the animals with human blood cells. So hematopoietic stem cells that when you deliver them actually into the liver of newborn pups, these stem cells, hematopoietic stem cells, can differentiate into all of the blood cell types you find in humans. So we give these immunodeficient mice a human immune system. Then we can infect them with HIV. And so that is what we did. And in order to disrupt osteopontin expression, we use something called aptamers, which are small pieces of DNA, which actually form a structure, a tertiary structure that can disrupt the binding of osteopontin to its receptors. And through a feedback loop mechanism, that leads to the downregulation of RNA expression as well as protein expression. And so we use this method by injecting mice with the aptamers every week, every other week. We could suppress and down-modulate osteopontin expression, asking is osteopontin required for HIV-mediated neuronal injury? So I want to stop here because if it's helpful or not, you let me know. I could bring up some slides, or should I just continue to describe the results?
I think slides would be great, actually. I think I just enabled it.
Okay.
I don't know about anyone else. I'm a visual learner.
Okay, great. So let me just bring up the pertinent slides here.
I like the colors.
Okay, awesome. Okay, great. So let me go to slideshow. Okay, just so you can see the overall experimental design. So these are the mice. I'll just use the short term. They're called NSG, but essentially they don't have an immune system. And we first conduct a whole body irradiation to actually deplete the bone marrow cells that the newborns have. Then they are injected again into the fetal liver, which is the first site of hematopoiesis. And then those cells will differentiate into the human target cells. And after about 4 months, then we can infect the animals with HIV. We would sample their blood, and by flow cytometry, we can measure and identify that the mice have developed a human immune system and have monocytes, B cells, T cells, etc. And then here we do the weekly aptamer injections. At necropsy, we harvested most of the tissues. Some we stored for later RNA analyses. We do quantified viral load, as well as the efficiency of osteopontin knockdown. So we had both female and male mice represented, and we had 4 groups, either animals injected with buffer, suppressed for osteopontin or not, or infected with HIV. Some of the outcomes that we looked at, we did behavioral studies to look at anxiety and locomotor activity. We also looked at microglial activation using neuroimaging, which I'll show you some results from that. We also plan to do immunohistochemistry to look at microglial activation, neuronal injury, and astrocyte activation. So I show you here in this slide, it is known since HIV, we've been with us for over 34 years or so. So we do know areas in the brain where virus can accumulate. In this nice review article here, you can see these small red dots showing the different areas, the thalamus, different areas of the cortex, etc., where HIV is known to be in abundance. And so we used a technique called RNA-scope. Maybe some of you are using that in your studies or have heard about it. And it's an in situ hybridization technique, which allows really sensitive single cell detection or single level detection of RNA. In this case, we're looking at HIV viral RNA. That's in the red stain here. There's some controls. This is in the spleen. So virus replication in the spleen, is much more abundant. So we can see here the viral particles in red in the spleen. In the brain, we could find some virus in brain tissue, was a very low level, very infrequent, but you can find infected cells in the brain. And this has been reported prior. And so how effective was osteopontin knockdown in the brain? As a proxy, just to make sure that the aptamers were working, you can see here in G, this is looking in the gut. And this is gut tissue immunostained for osteopontin. And so in this case, these were animals that were injected with a mutant aptamer. So that is an aptamer that had a mutation in the binding site. So it did not disrupt osteopontin RNA or protein expression. And this is quantified here in H. And so you see here, we saw significant decreases in osteopontin expression in the gut. In the brain, we used real-time quantitative RT-PCR. This was osteopontin normalized to GAPDH, a housekeeping gene. And while the results were not significant, we did see a trend of decreased osteopontin expression in the animals that received the functional interfering aptamer. Yes. So in terms of looking at microglial activation, one of the techniques we can use is positron emission tomography. And for this, we collaborated with a group at our institution, which has been a pioneer. This is Marty Pomper Laboratory, which they've been a pioneer at looking and developing radioligands for the translocator protein. Sorry, I don't have that spelled out here, but that's what TSPO is. And the translocator protein is a protein that is increased in expression on microglia that are activated. And so this ligand, it has a very short half-life. So it has to be made, synthesized fresh each time. So this is me actually waiting to receive the ligand. And then I had to run.
So, but I'm taking like 2 gap years before applying. So right now I'm like applying to like gap year positions.
Is there someone that's unmuted?
Yeah. So I've had like interviews, but I haven't like.
Okay, I think that did it. Sorry about that. Oh, I'm so sorry. You might need to unmute. I must have muted you in the process.
Mute. Okay, great. Excellent.
Sorry about that.
Not a problem. Okay, so I was saying that the radioligand for the translocator protein, which is elevated on activated microglia, has a very short half-life. So it's made fresh each time. For each time we had to do the imaging, we could image 2 mice at a time. And so basically, you know, this is a capability we have, but it is quite an arduous exercise. And I did get my exercise running from the hospital to the basement where the animals were there, where my postdoctoral fellow and the technician had the animals ready to receive the label. And so these are the kind of images that we can get. And you see here in the buffer injected animals with normal osteopontin expression. We're looking here at the brain there. You can see based on the red, the heat map, that in the infected animals, but which had less or reduced osteopontin, we saw more uptake of the radioligand. So this was actually counter to our hypothesis. So again, these are HIV infected animals with osteopontin. This was 45 minutes after imaging. We can see here the brain, but in the animals that had less osteopontin knockdown, had more inflammation, suggesting that osteopontin actually has a protective role in ameliorating inflammation. On the left here is showing time versus the uptake of the radioligand for the 4 different groups. And you can see here over this time activity curve that the infected animals with reduced osteopontin indeed had more uptake. This is looking at the whole brain. And again, so the important aspect is infection. So infection itself does lead to some inflammation. And this is even exacerbated when osteopontin is not there. And we saw also a linear correlation with viral load. So the higher the viral load, the more uptake of the label, suggesting the more inflammation. And we looked in several brain regions. And if you recall, remember that diagram I showed you earlier about the brain regions where HIV is known to accumulate. We know the cortex is intimately involved in some of the pathology, but also some of the neurocognitive effects of HIV. And interestingly, in the olfactory bulb, we also saw significant uptake. And then we went on to correlate. So of course, as you're doing in your own studies, you want to have, several ways to confirm an observation where you can show the same kind of phenomena using different types of experimental approaches. And so we asked, can we detect a similar type of difference by using immunohistochemistry? And so we did double labeling for the translocator protein, which is in red, and in brown is for a marker called IBA1. IBA1 is a well-known marker that is increased unactivated microglia. And so you can see some images here from the cortex and from the midbrain and some blow up of these images. Interestingly, we did find some alterations in some of the animals, some alterations in the hippocampus, specifically in the dentite gyri. We see here in the infected animals with osteopontin knockdown, the gyri seem smaller. There was also accumulation of cells in these fissures. What this means, we don't know yet, but it's interesting pathogenic features that we found. There was abundant translocator protein expressed in some of the animals in the hippocampus, and even in the durovenous part of the meningeal layer. And we did see also some interesting pathology in the cerebellum, where we saw this disorganization of the Purkinje neurons in the cerebellum of the HIV infected animals with reduced osteopontin. Okay, that's just another picture of that. Okay, so let me just, because again, I don't want to know the balance or what balance you prefer in terms of having slides and discussion. So, right, because you've been in the Zoom and lecture all day. So should I stop there and take some questions?
I'm thinking, yeah, maybe Vivian and Shamar, I know we had a list of questions, but I feel like you might have answered quite a few of them.
Yeah, actually, I had a question about the organization of the brain, the one that you just mentioned of the dentate gyri.
Yes. Yes.
I was wondering if it's the presence of HIV that changes it, but you mentioned it's like the knockdown, like the lack of osteopontin. So I'm a little confused, but.
Yes, we actually don't know. This was just, some of the animals that had this feature, but we would definitely, need to investigate that. One of the caveats in terms of this model our animals aren't treated with antiretrovirals. So, but the viral load levels are very low. So we do suggest that this model reflects like low level, chronic, ongoing replication, because the levels of replication are not very high. They are, let's say, at least 2 or 3 orders of magnitude lower than what other investigators have reported or used this model. So although we didn't use antiretroviral therapy, our model is kind of a surrogate for that. So yes, we don't have currently any more data investigating that pathology in the hippocampus that you noted. Yeah, I think it's so exciting. This was my first foray into mice and doing in vivo studies, because most of my work has been in vitro with primary cell cultures. And so it's very exciting. And there's so many critical questions, but it's which ones and what to do, because we can always do a lot, but it's focusing on what's the most critical questions. Shimar? Yes.
Thank you. There was a part in the paper that discussed HIV infected people experiencing like varying cognitive impairment. Are these neuropsychiatric and cognitive impairments caused by HIV, or like by the drugs used to treat HIV?
Very good question. So it's in 2 parts. So yes, right, if people didn't have HIV, they wouldn't be experiencing the increased cognitive impairments that you can identify on neuropsychiatric testing. So I didn't bring up, but there are several domains of executive function, and through neuropsychological testing, one can test for so it's not my expertise, but there's Stroop tests, there's different types of memory tests that are conducted. And so often people are functioning, or they feel they're functioning fine, but actually when you do the test, you actually see that their performance is more than 2 standard deviations from, say, the control population. But because people are aging as well, they're aging with HIV, so we have these confounding comorbidities. So you can have the HIV, you have aging, and you could have a disposition to dementia. So we do need to make sure that that we're disaggregating properly, but these are the types of confounders. And then added upon that is the antiretroviral therapy. So over the years, over the HIV epidemic, the antiretroviral drugs have improved, because the first generations used to cause a lot of toxicity, even damage to the peripheral nerves. So those then are not utilized anymore. And with the protease inhibitors, which came about in 1996, those there was a portion of those that were later found to have toxicity. And again, now those are also not used. Those are the efavirenz. And so if you get to invite Dr. Bumpus, she's the expert on the drugs, on the treatment and the therapies. And so those drugs are not utilized anymore. Now we have the newer integrase inhibitors, which work really great at blocking virus replication and preventing integration, because that's really what we need. We need to prevent the integration of, say, newly formed viruses, prevent them from integrating into new cells. And the other opening or gap is actually having inhibitors that block the transcription, and particularly the transcription of the early genes. So, testing of these newer antiretroviral drugs for their toxicity continues, but with careful monitoring. So the physician should be carefully monitoring their patients, and if there's any indication that the patient's having some cognitive impairment, depression, anxiety, they will modify the treatment regimen. So excellent question.
I have another question about, I think there was a slide where you had like the levels of OPN in the gut and then in the brain. And I wanted to know if you could explain more about how, or like, does tracking it in the body correlate to any sort of increase or decrease in the brain? Or is that a good model to track it in the body and have that relate to how it's expressed in the brain? Sorry, that was really wordy.
Oh, no. No, excellent question. I think you've pointed out the limitations, right? And I discussed a little bit in the paper, because again, the blood, right? There's a separation between the blood and the brain, the blood-brain barrier. And very important, because since the brain controls everything, we do not want substances just to be able to freely pass. So it's a gated interface that regulates the trafficking. And so we don't know, again, because with neuroimaging, we could do a comprehensive scan of the entire brain, each tissue, right, and through the whole depth of the brain structures. But with immunostaining, we can only take a survey of a few slices. So there's a limitation at the level of technique in terms of immunohistochemistry. And even with the more quantitative PCR, so we could take a portion of the brain, right? We could sample the brain tissue, isolate RNA from that, and quantify for osteopontin RNA, and that's what we did. It's still only like 0.1% of the total brain. So we don't get a comprehensive assessment of the efficiency of knockdown. And so one of the limitations that we have in the HIV field, we don't have these nice genetic tricks and tools that you have in other fields where you can do, we would need cell autonomous knockdowns or knockouts, right? Where we could knock out specifically in the brain and not in any cells in the periphery, or vice versa. So there's definitely limitations. And so we would have to maybe do a more comprehensive, to answer your question about the effectiveness of the knockdown. We did see a trend. There was one outlier in that data in the HIV infected osteopontin knockdown, but there was a trend. But because of the overlap and the variation between the animals, it didn't reach statistical significance. But I think, the fun thing about science, and when you get a result, when you use a technique that is robust and you find something it is encouraging. So, so this study really relied upon the PET neuroimaging analyses and seeing that difference. Because actually nobody has, I'm sorry, no, so far, no, there have not been any reports as yet that HIV infection in these humanized mouse models leads to neuroinflammation. So it was by perturbing osteopontin expression that we actually saw what it's doing and how it's contributing, and that the virus is indeed causing inflammation. We didn't do a titration, right? So it may be in subsequent experiments, we actually need to give a higher dose of HIV, because the dose we used was much lower than some, maybe 10-fold lower than what other laboratories have used. But yeah, there's a lot going on when you do animal research. Yeah. Did I answer your question?
Yes, you did. Thank you. It was very cool.
Just keeping up with, sorry.
I'm so sorry. I just realized that I got a question in the chat, which I just wanted to address. Are different amounts of OPN required for different races? Because I guess Dr. Bumpus had found that 45% of amino acids have 2 copies of a gene that makes it so that they detox HIV treatment faster. Oh my goodness, AAs is for African Americans, not amino acids. I'm in biology mode, I'm sorry.
Oh no, you're good. So the question was, are osteopontin levels, do they vary by race ethnicity? I actually don't know. The study that we did in 2011, because we quantified osteopontin in the plasma and cerebral spinal fluid of a group of patients it was called the Northeast Atlantic AIDS cohort. And it was made up of individuals from Baltimore, from Chicago, and I think there was one other, maybe Philadelphia. So I think a large proportion of those individuals are African American, but I don't know if we have a comparison group. So I can't answer that question. Yeah, that's a good question. I've actually never thought about that, or if that could be potentially involved in pathogenesis. Is that why you were asking, the person who asked the question?
Oh,
But
it looks like she was just basing it off of Dr. Bumpus's research.
Okay. Yeah, I know Dr. Bumpus, in terms of when Shamar was mentioning drug toxicity. So she may have been, that replies to, the detoxification mechanisms. And so the mitochondrial, those CYP proteins, right? Which are so very, very important. Yeah, so again, why we need, very close monitoring of drug toxicities, because the drugs are not going to, can potentially and do differentially affect different groups. And hence the need for testing in, as wide a variety of people as possible, because, we know that about, right, blood pressure drugs. I know that from my own family, yes.
Thank you. I'll pass it back to Shamar.
Thank you. Just to keep up with timing, we were hoping to ask you some questions about your career in general. I guess we can start with what made you go into the field.
yes, I touched a little bit in the beginning, but really was in college, right? Making that transition from high school to college, I stumbled a bit and ended up reaching folks and folks reaching out to me to provide support. And in so doing, I found a mentor, and she was a microbiologist, that Catherine Atkinson, and she welcomed me to her laboratory. It was a yeast biology laboratory, and that's how I got involved in research. And it was a new world for me, a new exposure. But I was able to build up, that science identity. I enjoyed working in the laboratory, and it was, kind of one mentor in their network handed me off to the next. And so I did summer research during my years in as an undergraduate, in my junior year and senior year. And I ended up getting paired with Susan Henry, who was at Carnegie Mellon University. And she happened to be the head of the graduate school, former head of the graduate school at Albert Einstein College of Medicine. And I thought I was interested in immunology. And she said, hey, Einstein has a great program. And I applied and got in. And so, yeah, and then I fell in love with the macrophages, working with tuberculosis. And then I did a postdoctoral fellowship at the Aaron Diamond AIDS Research Center, which used to be across from Bellevue. They recently moved, well, they're right at Columbia now. So I don't know if you know Dr. Hope, Dr. David Hope, moved the Aaron Diamond AIDS Research Center, is now part of Columbia University. But that is where I did my postdoctoral fellowship under Cecilia Cheng Mayer. And that's how I got started in HIV research. And it was really, asking the question and focusing on HIV latent infection. And then the fact that microglia are the macrophages and the immune cells of the brain, that I ended up in the neuro HIV field. So it's been pretty phenomenal. I'm still learning, especially neuroscience. It's so exciting. And it's really exciting, where I'm bridging the fields of the immune system and intracellular pathogens and the nervous system. And so, as we know, you've probably have heard about, right? People are very interested in this. It's reached the general public about how viruses, right, like COVID, can disrupt homeostasis in the brain. Yeah.
I wanted to ask, so you're also the director of all these programs. So when did you get involved in translational research? And was there a transition process, or has it always been a part of your career?
Yeah. No, it was definitely a transition process, but I do think during my postdoctoral fellowship at the Aaron Diamond AIDS Research Center, many of the staff investigators were MDs doing research, but we also had PhDs, and there was collaboration between the two of them. And so there's that hypothesis testing from the basic science side, but then asking, is that phenomena present actually in people? Do we see the protein is increased, or the RNA is increased in the plasma, in the RNA? What do we know patients are experiencing that we can then ask, can we develop a solution or pivot to study that in the laboratory, in a controlled laboratory setting? And so I think that was definitely brought me closer. But I guess I have, when thinking about, my graduate studies with tuberculosis. So I have always been interested in pathogens and how they intersect with the host and the immune system, and how can we strengthen that so that all the time the host wins. And then at coming to Johns Hopkins, it was further amplified. So it just so happened that the person I came to work with at Johns Hopkins was located in the Department of Neurology. And so it definitely took the translational research to another level, because of course, the majority of the faculty are MDs, but there's some who are MD PhDs, many who are MD PhDs. And again, that collaboration is very tight. And there's a very close and even intentional focus on translating. And we do have a large, just I'm sure you do at Columbia as well, a large number of faculty who have the experience of actually taking their discovery from the bench to the clinic. And some have been successful. So it is definitely very, very exciting. I do appreciate being able to be in this space. Yeah.
Can we ask what your thoughts are on the grad school application process? Would you encourage students to do it? Is it not so necessary?
You're saying just that last point, it's not necessary in regards to?
I guess intention matching, purpose. What would you say to someone who's contemplating applying to grad school if they're not necessarily completely certain about the choice?
Okay, wow. I hope everybody on this call is applying to grad school. Okay, we need all of you. It's been so difficult, right? Students are asking for faculty who look like them. And it's more just not the looks, right? It's to know that we all have a role. We all are needed, that your perspective matters, that you bring something unique because of your background, where you've been raised, what you've been exposed to, yet or not. You bring a unique perspective. It's not just words. I mean, I don't know how to say it, but I actually experienced it, right? Because a lot of times, right, we know there's competition in research, right? But I found this protein. I didn't know at the time that it would be something I'd be working on. Gosh, it's going to be 10 years. And osteopontin, when you look in the field and you type in osteopontin, all of this literature comes up. A lot of it is in the cancer field. And people say, oh, osteo, isn't that something with the bone? Yes, because osteoclasts are the macrophages of the bone. But there's something unique, right, about my perspective, my insight, based on all of the research experiences that I have had. And, sometimes I wonder about that myself. But it is that building of that self-efficacy to ask questions, to be curious, and then want to go to the lab and do the experiment. I still have to work in the lab. I love it. But, I'm at a phase where, okay, people moved off to graduate school, to second postdoc, to medical school. So it is, if you have the passion for discovery, you want to know why, and you love getting up and going to the lab, it's for you. And so, choose the lab carefully, right? Because there will be lots of failures, but look at those failures as that is not the answer. Okay, there's something else. There's something else. And ultimately, in collaboration, you talk amongst your colleagues, with others, you dive down into the literature, you will ultimately get the answer or a little piece. And things are moving so fast now, and this just with the technology, right? I'm sure all of you guys are, doing different types of sequencing. Now we can do single cell sequencing, looking specifically in nuclei. So it's a very exciting time. And I know often for those of us that come from, backgrounds where, you're the first generation, your family doesn't understand and like, you should get a job. You got all this debt, you got to work, right? And that's very, very real. Maybe you might have to take a detour and you have to work for a little bit, excuse me. But you can work in a lab. Maybe you can be a technician for a while, stay close to the research, or investigate in industry position. If you want to stay close to research, but you need to, take care of some of the, there are financial constraints. Those are very real. But be open to talk with your mentors about these issues, because they're challenging, right? But you can pivot, but you may not, it may not feel comfortable at the time, right? But just know you can always pivot and change and still get to where you want to get to. It's not always a linear pathway. You just got to try it, see if you like it. If you don't, okay, switch. And don't worry about the time. Some people want to like, okay, I got to do this in X years and I got to be here in X years. And, maybe with this COVID environment, right? That is affecting us and changing things. Okay. But you'll get there. We're a little bit delayed, but you will get there. I kind of went on, but did I answer your question?
Yeah, I definitely think that you did. And I just wanted to ask about, well, you mentioned how, we need more underrepresented students in STEM and STEM research. And I know that you're the director of a bunch of different scholars programs, which I think are for high school students. I'm not quite sure, maybe both high school and undergrad. But how did you start that? And how do you balance doing that and doing research?
Yes, thank you. Yes. It's certainly not easy, but I think as you just observed, I'm very passionate about it because I know the impact that it had on me for somebody to take an interest in me. Okay, and I wasn't perfect. Maybe even some of my faculty mentors were saying, oh, she was hard-headed, she didn't get it. It took her so long. But they invested in me. And so I in turn invest in others. And so the high school program was actually, there was a faculty member who was in an informal way inviting students to her lab. And then a couple of years later, she had to pivot to be vice chair of research. And so our director reached out to me. And so we then kind of had a more formal program. And I actually even got funding from the NIH to run the program. And we had a very close philanthropist, very close collaboration with Suzanne Cohen, who was really pushing and is a great supporter for young people in Baltimore City. And so she was the fuel and the fire for this program, the Johns Hopkins Internship and Brain Science Program for high school students. They get paired with one of our faculty. They get to do research, either clinical or basic science, depending on the faculty member. And it's a transformative event. In fact, today I just got the list of the students that were accepted to our Johns Hopkins Neuroscience program. And one of the students is on the list. She was in our high school program. And I guess they figured it would be a conflict of interest. I don't know if she wanted to surprise me. She didn't even let me know. I sent her an email. I'm like, well, Chanel, I hope you come. But it's exciting. It's making a difference. I do have a publication to get out on that because we did do an evaluation. And so as you mentioned, the balance. And of course, we hear that, right, that faculty of color are often, asked to do this, people call it service work. And but for me, which I tell my students, I don't think I would be here if I wasn't doing that work. Because the students were looking at me and I'm like, wow, if I fail, what are the students going to say? What are they going to think? They're going to think, well, gosh, if she can't do it, I can't do it either. No, I do it. I did it, so you can do it too. But you're going to do it. You guys are going to do even better. So I work very hard. I think in terms of the research questions, obviously I cannot do everything. So I have to be very strategic about the questions that I do answer. I think I've focused on a system that maybe is more difficult. So it's, not something a lot of people can do easily. So one does have to think about, your niche. So I've created this niche that has allowed me to thrive. I think, in the beginning, it's always hard when you're learning how to write grants. And I would say, when I was coming up, there weren't programs at the different stages, particularly at the postdoctoral fellow stage is a very critical stage. And if you don't have the mentorship where people will share their grants or they'll read your grants line by line, give you the feedback you need, that is the tough part. So that continues to be a problem when we look at the funding rates for the NIH funding rates for the R01 grants for faculty of color. It continues to lag severely behind. But the NIH, I would say the last, probably 7 years, they've been on top of this. And so there are programs that address that. You can be early career faculty and be allowed to sit in on a panel so you can see how grants are written, how they're discussed. And there's also grant writing workshops so that you can get that experience. Because if you want to be academic, in the academic environment, the National Institutes of Health is the major funder of research. So those kind of things are very important. But I think the push now, and hopefully you all are experiencing that, excellent mentorship, and you should have a team, right? Because not one person can do everything. So definitely, reach out widely for mentorship and have a team. And I would say one other thing, make sure your next steps, whatever it is, you also are the interviewer. You're being interviewed, but you are the interviewer, right? Because you want to make sure that it's a place, environment in which you can thrive. So yeah, I could talk more. I'm sorry about the undergraduate programs, but but was that fine? So we target underrepresented students. And I have a collaboration with a colleague who's deaf, hard of hearing. So it's very interesting. Deaf, hard of hearing individuals from deaf, hard of hearing backgrounds are also severely underrepresented in STEM, and they face some of the same types of questioning that people of color do. So I've been learning a lot from my deaf, hard of hearing students who are in our scholars programs.
That actually makes me particularly excited to hear that, because I'm a child of, like, I'm a coda, but both of my parents are deaf. So yeah, they definitely get questions, but they're not nearly at a, that sort of a standing. So
Yes.
different kind of questions. But I wanted to ask you, actually, is it possible to be overqualified when it comes to a career in research?
I don't know. Give me an example. I don't think I've ever heard that. Overqualified.
I suppose, like, I guess if you're looking at job applications, and they usually say like preferred masters, preferred bachelors, preferred whatever. And I've heard that sometimes, like, they won't want someone that has a PhD or more education because maybe they have to pay them more. I'm not sure. I don't know if that's something that you've heard specifically within research, though, or
well, it could, in terms of, so those positions, that sounds like more industry, where perhaps that is a factor, and it's probably related to the pay. I can't say, I haven't been looking at advertisements in industry and whether are PhDs having problems getting positions. So you're speaking right after you complete your PhD. So there are postdocs. So many pharmaceutical companies do have postdoctoral fellowships within the company. So that is an option. And often, if you want to go in industry, that is a good way to segue to that pathway. So it may mean targeting particular companies, and a willingness to move where those companies are. Yeah. So yeah, I know what you mean, but talk to your professional development office. Because yeah, I mean, I think you'd want to use, you're in a PhD program, you want to use your PhD. You don't want to feel that you're overqualified. I think I didn't even like that term.
I know.
Because what do you want, a robot?
Yeah, it's something that I've heard a lot, though, like when people are considering higher education. So I was curious to see what your thoughts were on that.
Oh, so they may not even plan to pursue PhD because they see the positions and it says, hey, masters or etc. I see. Yeah, I'm not. So, that's a question of the skilled workforce and what is needed, right? Because we always have to be looking, short, mid, long term. So what are the needs in 5 years, in 10 years, in 20 years? So the academic workforce, I was looking something, the AAMC recently, and I don't know if it was 55%, don't quote me. It was either 46 or 55% are like 50 and over, right? So people are going to be retiring. So we do need to fill filled those job positions, but I'm not totally, up to speed with those actual numbers. But I think that is probably the way people should look at it. But then passion, but that hurts me to say, oh, you're overqualified. But yeah. Thank you for asking. I'll hopefully next time be more informed about that.
No worries. I'm thinking that time-wise, I should probably open it up to the crowd. If people have questions that they'd like to ask, feel free to type it in the chat, or you can unmute yourself and ask.
I have a question, just another general question, but I guess would you like to talk about any challenging moments in your career and like how you overcame them? Basically, how to have the strength to keep going, and being an undergrad right now, especially because of COVID and Zoom learning and all of that stuff is really hard. So I guess any advice on how to keep going forward?
Yeah. I think, definitely community. So having my peers, they were often going through the same kind of challenges. That would be like when experiments didn't work. So knowing that I wasn't the only one struggling with getting my experiments to work. So I think that is very important to have, express some vulnerability and willingness to share, how you're feeling with your peers. So that it's not like, oh, everything is okay for them, or they seem to be happy and everything seems to be going well. Well, no, they may not just be sharing, the difficulties that they're having. And yeah, make sure you have some other activities, take a break. If it's something that's getting you really down, maybe take a break from it for a bit. Go do something fun. Eat your favorite food, ice cream, chocolate, whatever it is. And feel better. Talk to your family, because your family is like, oh, you're so great. You're at Columbia University. We're so proud of you. Right. And it's true, so they look into you. So know that the struggle is part of the process, and that it makes you stronger. So, there were times, I would say it's only been maybe 4 years ago, that I could talk about how, I had miscarriage. I was in New York there and doing my postdoc, and I had a miscarriage, right at work. And not only one, but two. Then I had to go third one on bed rest. But there were years for many years, I couldn't talk about that. But, we tried. Now you heard I have a son who's doing great. So those setbacks, those challenges, at the time you can't feel it. It doesn't, of course, you feel bad. And you must go through those necessary emotions. But once you come out on the other side, you're stronger. And then you can share with others. And then that helps other people. Yeah.
Thank you for sharing.
You're welcome. Yes.
Hi. I'm actually a bio teacher at high school, at a high school in New Jersey, and I teach freshmen. So what I'm trying to do is to kind of integrate all students into my class, and kind of use disaggregate instruction, where we're taking like the big concept and kind of just trying to simplify it and use more simple language, stuff like that, so everyone feels included, right? So, but as I'm looking at your research, and generally when you get into more complicated research and in college, the language gets very, very complex, I would say. Do you think that there's a way to maybe simplify it or kind of, I don't know, make it as simple as possible so that more people can learn, or if that's beneficial? Yeah.
You're right on, Merrick, because we do a disastrous job of explaining our work to the public. And I definitely need practice. And yes, definitely it can be explained more simply.
Mm-hmm.
And so I would love, hey, if you want to invite me sometime by Zoom, I would be happy to practice, to practice beforehand. And then you could give me the feedback about how I did, because this is so important. We all see, you know, in this last year with the pandemic, that people don't understand. I had relatives reach out to me. They wanted to understand more about the COVID vaccine and how it works. Because it was like, well, why could they do it so fast? And I had to explain to my cousin that this was research that was going on for years, that they're capitalizing on, you know, what we do, what scientists do every day, but nobody knows. The general public doesn't really know about us. They don't see us. And when we do try to explain, it's all filled with jargon. So no, you're right on. Yeah.
It's definitely, I don't even know how to sometimes. I'm glad like I teach ninth grade, it's doable, right? But then when it gets way more complicated, I'm just like, how am I going to get this information out there? So I was wondering if that was kind of possible when it comes to college.
Yes. And then on the flip side, right, because I've interacted with the high school students, and all the students, they know nothing about the topics that they're studying. And they are able to dive down into that literature. And with some, you know, practice, we go around every week, like I would have lunch meetings with them. And they would talk about what they're doing. So at the beginning, you know, there's a surface level understanding. And then each week it goes deeper and deeper. So they can rise to the level, but it is combined with hands-on experience. So they're able then to connect what they're doing in the laboratory and then actually understand what is in the paper. But it's no excuse. We as scientists definitely need to be able to speak to the public in simple terms, in good analogies. Those that have really good analogies. And there's some great examples out there, like the astrophysicist. You know who I'm talking about. But I listened to him a couple of weeks ago. Simon Tyson, Neil Tyson. That's right. He's terrific. Yeah.
He's really good at that, actually. He's really, really good with that.
Yes. Yes. Yeah.
Mm-hmm.
Thank you. Thank you. And thank you for what you're doing in inspiring young minds.
Oh, thank you too. I appreciate it.
You're welcome. Hi, Sophia. How you doing? Sophia.
I'm doing well.
What do you?
Oh,
yes. What are you studying?
I'm going to be a neuroscience major, and I'm probably going to minor in Spanish.
Muy bien.
Yeah. So I don't know. I've been trying to think of a question, but I feel like everybody else already covered everything, and I'm just like, I don't know, sitting here in awe of like your work and like your passion for your work. It's so incredible, and especially how you help like other like underrepresented people and like enter academia and like continue on their journey. And it was really interesting to hear about it because like in the future when I'm like established in my career, because well, I want to be a doctor, I want to be a pediatrician. I also like want to do the same thing to like help other people enter the world. Yeah.
Awesome. Thank you.
So actually, do you have any like tips or like how did you get into it and how did you continue on?
Mhm.
Yeah.
A really good way is summer research programs. So stay engaged. And even there doesn't have to be a program. You can directly, if there's a professor, their work that you are inspired by, and you can go directly to them, talk to them, say, hey, you know, I'm interested in doing research. I really like your research. I read your paper, and this sounds really interesting. Would it be possible for me to do research in your lab during the summer? And hopefully, maybe they might be able to pay you because we know that, you know, we all need to work. And so hopefully they can pay you. And if they can't, there are other programs out there. So you could reach out to me because especially with this virtual environment and there's differences in universities allowing in-person training. We will have mechanisms whereby, you know, some of our trainees may stay at their undergraduate institution and do research, and then we can pay their stipend. So just another reason to build the network, to know what is available, and just to ask, because you know, faculty and people, they definitely want to help. But don't get discouraged if they say, oh no, I don't have this time or I don't have space. Then you can ask them, well, can you recommend somebody else? Do you know another person who might? Yeah. And so just try to stay positive. You know, you may get 10 rejections, but then the 11th one says yes. Thank you so much.
Oh.
You're welcome.
Does anyone else have any questions?
Hi, Nada, Alex, Juliana. It's your chance.
Well, it's a very good point. You only need one person to say yes, really. The worst they can say is no. I suppose with that being said, Dr. Brown, thank you so much for taking the time to chat with us. And I'd like to thank our audience for attending our talk as well. I hope you all enjoyed it. And this one was particularly interactive, so I'm really, really glad.
Awesome.
Please follow us on Instagram and Twitter. I will post it in the chat. And if you subscribe to our mailing list, you'll be able to see our next events.
Awesome. Well, thank you so much. It was a pleasure. I appreciate it. I wish you all continued success in what you're doing. I put my email into the chat. You can reach out to me. I'm part of your network now.
Perfect. Thank you so much.
Welcome. Bye-bye. Stay well. Get your vaccine when it comes. Right? Yeah. Okay. Bye.
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 Bye.